Hi, good morning, everyone. It is my pleasure to introduce our first speaker, Dr. Michael Levitt from Stanford University. He is a pioneer in the computational structural biology. As many of us might know, that in the 1940s, that scientists already understand that proteins are strings of amino acid pearls, and they are very important building blocks and enzymes. So with the application of X-ray diffraction in biology, scientists began to understand that the necklace of the pearls actually formed this particular alpha helix structure, which we now know is the most prominent structure in proteins. And in 1953, the double helix structure of DNA has also been discovered. And in 1961, the first X-ray structure of protein, the structure of myoglobin, has been discovered. And you can see the interaction of this ion with all the amino acid residues in this protein. So people at that time began to realize that biology is all about detailed molecular interactions. When we talk about molecular interactions, we have to talk about a chemical bond. A chemical bond is the most basic unit of molecular interaction. When we are in college, when we study the chemical bond, we were told that a chemical bond is like a spring. If you stretch or squeeze a spring, it always tends to bounce back. So it is like a chemical bond. If you translate this model into an energy barrier model, we understand that a chemical bond is relatively stable, and it's sort of vibrating at this pit of the energy barrier. If you can overcome this barrier, you can actually break a chemical bond. So here's a side story. When I try to search bond, all I got is James here. So you have to really be particular. You have to type in chemical bond, but not only bond. So it is relatively easy to simulate a chemical bond with a spring model. But in reality, in real life, our proteins are very complex structures. It is formed by thousands and even tens of thousands of chemical bonds. So how can you turn these static, like 3D crystal structure into these live dancing molecules? So for me, it is very difficult, and I believe for many of us here, we don't really understand how to do this transformation. But obviously, these guys know what to do, and they have been working together to develop this complicated computational model to simulate these live molecules. And due to their seminal work, these three scientists, Dr. Kaplas, Dr. Levitt, and Dr. Warshel, shared the Nobel Prize in Chemistry in 2013. So today we are very lucky to have Dr. Levitt here with us, and he's going to talk about the simulation of functional motions in macromolecules. And Dr. Levitt?